Today I'm going to talk about CRISPR-Cas9, as Abdullah announced. CRISPR-Cas9 has been called a gene scissor, so a system that can precisely edit our DNA and make very targeted changes in this DNA. And as Abdullah also mentioned, there's been quite a bit of hype around CRISPR-Cas9. So much so that if you look at how many publications mention CRISPR in recent years, you just see an explosion. In 2002, it was first mentioned in one publication. Last year, we had a bit more than 3,000 publications mentioning CRISPR-Cas9. This year, we're on pace to surpass even that. But CRISPR-Cas9 is not just something that is in research and that research publications are written about. It's also something that starts to hit medicine and starts to approach things that really will influence our lives. For example, in 2015, a team of Chinese researchers for the first time changed the DNA in non-viable human embryos. So those were embryos that could never grow to be full humans, and there were some technical difficulties in the studies, but they managed to do some first edits. And then just 2 years later, a second team of Chinese researchers managed to successfully cure a handful of viable human embryos of a genetic disease. So just last year, this technology was used to change a human embryo that could technically grow to become a full human. But CRISPR is not something that can just be used to mess with embryos. It has a lot of other application or potential application in medicines. Just this year, multiple clinical studies, clinical trials have been announced that will take place. One of them to cure sickle cell disease, and a second one to treat melanoma by changing a patient's immune cells. But, so summarized, we can say we have a fascinating technology here, and there's a big hype around it. But I don't really want to talk too much about where it's going, but more about how did we come here? How comes we know about CRISPR and know how to put it to use for all of those things? And what I'm going to tell you this evening is a story in 3 acts. Act number 1, sequencing in the late 80s and early 90s, and scientists observing something interesting in bacterial DNA. Act number 2, a bacterial immune system and how we came to understand how it works. And act number 3, precisely cutting DNA and all its potential applications. And through this evening, I'll guide you through how we came from this to CRISPR and editing embryos. So, Act 1, sequencing. The year was 1987. The Berlin Wall was still standing, and DNA sequencing was in its infancy. A group of Japanese researchers were sequencing the DNA, shown here as those black lines, of a bacteria called E. coli, shown here in blue. And they were specifically just looking at one of the 4,500 genes of E. coli. The gene they were looking at is this YUP gene, and they were sequencing the gene itself and a bit of the region around it. In total, they were sequencing 1,700 base pairs, which is less than 5% of 1% of all the DNA in the bacterium, from which you can see that this is not to scale. There's much more DNA in here. They were sequencing this DNA just to understand how this gene would work and how the protein would work that the gene makes. And they had some funny side observation, something they were not looking for but was there as well. Next to this gene, they found some short fragments of repeating DNA, so some short repetitions in the genetic code. It was unclear what they were, and I quote the last sentence of their paper to summarize what they thought about this. So far, no sequences homologous to these has been found elsewhere in prokaryotes, and the biological significance of these sequences is not known. Or in other words, we also don't know what we found here, but it kind of looks interesting. Well, at this point, this finding could just have been ignored, or it could have been studied further. And the same group continued to study those repeats, and just 2 years later they published again and could show that, well, it's not just this one bacteria that has those repeats, this is actually something more common. Multiple different bacteria have these kind of repeats in their genome. They then published this and showed that you have a repeat of a sequence and you have something unspecific in between. No idea what this is. You have the same repeat and something, a repeat and something. You can have this multiple times. You have multiple repeats of the same kind of fragment and just something in between. And they were not the only group finding these kind of repeats. I'll quickly take you through a story through the 90s of how different groups found these kind of repeats in bacterial DNA. First, in 1993, a group found those kind of repeats in the bacterium that causes tuberculosis. Then, in 1995, another group found these kind of repeats and made some first speculation. Well, maybe they're involved in the replication of the bacteria. In 1996, they were shown in yet a few other groups of bacteria, and it could be shown that those sequences, those repeats, are actually very well preserved. In 1999, a first application was found for how to use these repeats for something. You could use them to tell different bacteria apart. And in 2002, this whole phenomena finally became got a name, and this group named it CRISPR, which stands for clustered, so close together, regularly interspaced, so always with something in between them, short palindromic repeats, so some specific kind of repeats in the DNA, or CRISPRs. And they also found some genes that always happened close to those CRISPR sequences, and they called them CRISPR-associated or Cas genes. So now we have a funny phenomenon, we have some repeats in the DNA, we have a name for it, more than a decade's worth of research, don't really know what to do with them. It's not necessary that this has to be pursued. But there were more studies coming on these kind of repeats, and in 2005 we come to the second act of the story, the bacterial immune system. Because bacteria don't live in a vacuum. Bacteria live around viruses. Viruses that want to go and kill the bacterium. And the bacterium doesn't really want to be eaten or killed by those viruses. So what the group found in 2005 is that so far we've always focused on these red repeats in the bacterial DNA. The actually interesting thing is probably the thing in between them. Because what's in between them is DNA that is from those viruses. So, well, it's kind of interesting that there's DNA from those viruses in there. The really interesting thing is that bacteria that have a DNA of the virus then are not susceptible anymore to the virus. So if this bacterium has this pink DNA fragment from this virus cannot kill the bacterium anymore. So it seems to be some kind of immune system going on here. But how could that work? We have an immune system, but it has specialized cells that patrol our body. A bacteria is just one cell. This one cell is much smaller than even one of our cells, and still it seems to be able to somehow fight viruses. So it became an interesting area of study to try to understand how this immune system actually works. In 2007, there was quite big progress made in understanding how this immune system worked, because this research group here could show that for the bacteria to become resistant against those viruses, they need to meet those viruses once, then they can get this DNA into their own DNA, and then combined with the Cas proteins, so those CRISPR-associated proteins that are always next to these sequences, they can actually fight against the viruses and have their active immune system. And over the following few years, it became clearer and clearer how this process actually worked. So yeah, and if the immune system works, they kill the viruses. Over the next few years, I just picked a handful of studies that showed what happened. The first study here in 2009 was very fascinating because they could show that the way this immune system works is that the Cas9 protein, together with the specific RNA, can cut up other RNA very specifically. So you can go to one specific sequence and break this. Just a year later, a second group showed that in bacteria, the Cas9 protein would take one of those sequences from a virus, search the other viral DNA, and break the DNA of the virus. So the way the immune system works is that the bacteria has a memory of what kind of viruses it has seen before, and if it finds them again, it breaks their DNA. And in 2011, a third study could show how the bacteria would actually take this DNA from in between the repeats and bring it to the Cas protein so it could go and search for viral DNA. So now we're at the point where we found some funny repeats. It was actually interesting viral DNA in between them. What did we learn from that? We learned that what we observed here was a bacterial immune system. Well, a bacterial immune system is interesting, but not really directly applicable to us. What should we do with a bacterial immune system? Until a very seminal study in the field came in 2012, where they showed that this doesn't only work in bacteria, this also works if you take it in any other kind of cell or any other kind of system. You can take this Cas9 protein, give it a guide RNA, and this then searches for a specific sequence and breaks this. To give you a visual idea of how that works, there's a very nice video made by Nature Methods that shows this exact process. What you can see here coming up is the Cas9 protein, so the actual form of this protein that is bound to a guide RNA, so an RNA that gives a target. This complex, they form a complex, the Cas9 protein and the guide RNA, and this complex can then go around the cell and search for other DNA, especially search for viral intruder DNA, and it can bind to DNA at specific places, can unbind the DNA, so open it up, and check whether the sequence that it has in its own is a perfect match with the sequence of the DNA. And if it finds a perfect match, if it finds, yes, this is viral DNA, it can then break the DNA, and thus, in the bacterial immune system, break the virus. And the cool thing of this is that, well, it works for bacteria to fight viruses, but it also works for many other things. Once it was shown that this is a generalizable system that very easily targets DNA, the number of publications exploded, and a lot of very fascinating studies came out. For example, in 2013, this one that showed that we can actually put the Cas9 protein in human cells, give it a target of where we want it to do something, and it will then go there and break the DNA there. And also in 2013, another study showed there's a way that we can not just break DNA, but we can determine what will actually be inserted in that part of the DNA by using a process called homology-directed repair. We can give a template for how the sequence should look afterwards, so we can guide our Cas9 somewhere, let it break the DNA, and insert a new sequence that we wish in this place. This was a story in 3 acts. So to summarize, what we've seen is we went from sequencing bacteria in the late 80s and early 90s and finding some funny repeats in their DNA, to understanding that those funny repeats actually were a bacterial immune system, or part of a bacterial immune system. This bacterial immune system allowed bacteria to fight against viruses. to then making the leap that we could use these same kind of proteins and this same technology and put it in other cells and have a genome editing tool. A genome editing tool that can speed up basic research, but that can also be potentially used to change embryos or to find new kind of treatments for genetic diseases or for cancer treatments. What is the moral of this story, to finish up? The moral of the story is that CRISPR-Cas9 is just one example of a technology that was found through decades of basic research where we didn't know that we would end here. We didn't know that in the end of sequencing bacteria we would find genome editing technology. It was decades of basic research pushed by curiosity and the willingness to try and understand the system. And the cool thing about this example is there might be many other things out there that we're just now starting to study that may end up giving us tools of similar impact. Thank you very much.